And so previously we discussed the organization of muscle cells as well as the basic molecular process of muscle contraction in the sliding filament mechanism. But there's also a role for calcium in muscle contraction as well. And we're going to discuss that here. Right, and so we talked about in the sliding filament mechanism, there needs to be a signal to contract received before myosin heads bind ATP and the whole thing kind of progresses from there. And that signal to contract in a muscle cell is received from neurons, specifically motor neurons, right? And when we refer to kind of this motor neuron as well as all of the muscle fibers or muscle cells that it innervates, we're referring to a motor unit, right? So you can see the motor neuron here in yellow, the muscle cells it innervates, or this muscle fibers, as well as what are called motor end plates, which is basically the connection points between the motor neurons and the muscle cells, right? And neurons make contact with muscle fibers at a very creatively named place called the neuromuscular junction, right? The junction between neurons and muscles, right? And each muscle fiber only has one neuromuscular junction. And this is helpful in kind of controlling the signals that are received, right? Because you only really want to be receiving signals from one neuron. Otherwise, it might get confusing for your muscle cells, figuring out when to contract and when to relax. Okay. And the number of fibers or muscle fibers that are innervated by a particular motor neuron depends on the type of movement, right? So there's only one neuromuscular junction per fiber, but one motor neuron can innervate, as you see here, multiple different fibers, right? It has multiple processes, comes out, and it might be innervating anywhere from 1 to 2,000 different fibers. And this number of fibers that are innervated by the motor neuron, as I said, is correlated to the type of movement. So typically with fine motor movements, like using your hands to write or do something that's kind of highly dexterous, one motor neuron is only going to be innervating several muscle fibers, maybe a couple hundred, right? Because there then sort of becomes this level of control that motor neuron has, and its signal is dispersed over less fibers, right? And so each fiber can respond a little bit more carefully to a signal received from that neuron. Whereas a motor neuron that innervates thousands of muscle fibers might be controlling a gross movement, a large movement of your arm or leg, or something that would maintain your kind of posture and stability. And that's because you're diluting that signal sent from a neuron into thousands of different cells, right? So they can't really respond quite as acutely and finely as they would if there was less cells innervated by that same motor neuron, right? And so what actually happens to communicate the signal from a neuron to a muscle cell to contract? Right. That happens, as we said, at the neuromuscular junction, or the meeting between motor neuron and the muscle itself, right? And so neurons will fire an action potential or kind of an electrical signal that needs to be converted into a chemical signal to be received by the muscle cells, right? And so that chemical signal happens in the form of release of a neurotransmitter or a small molecule called acetylcholine. So an electrical signal coming from this motor neuron triggers the release of the neurotransmitter acetylcholine from the motor neuron, and that acetylcholine then binds to receptors on the muscle fibers or the muscle cells. And that binding of acetylcholine causes the electrical signal, or that impulse, to travel down the muscle cells and into what a specific structure called transverse tubules, right? And so the muscle fibers are here, and they are groups of many, many myofibrils. And so the signal is received by the fiber and then transmitted into the myofibrils themselves by the transverse tubules. And the way that the myofibrils really respond to this signal from a neuron, or this action potential, is to release calcium ions from the sarcoplasmic reticulum into the cytoplasm of the muscle cell. Right, and so the sarcoplasmic reticulum is basically the ER, or the endoplasmic reticulum, but of a muscle cell. And as we all know, the ER stores calcium, right? And so once the signal to contract is received and transmitted down transverse tubule, that action potential triggers release of calcium into the cytoplasm. And then calcium has a specific role in the sarcomere. And we already talked about how myosin heads bind to actin in order for the kind of sliding filament mechanism to take place and for contraction to happen. But there's a specific site on actin where myosin can bind. It can't just bind anywhere, right? And usually those binding sites are actually covered up by this kind of rope-like structure you can see here in purple, which is known as the troponin-tropomyosin complex. And that troponin-tropomyosin complex kind of makes a rope, as you see here, that wraps around the thin actin filaments, blocking the myosin binding sites and preventing muscle contraction. However, when calcium is introduced into the equation, calcium actually binds the troponin-tropomyosin complex and untwists it, revealing actin or myosin binding sites on the actin and allowing those two molecules to interact with each other. Right, so calcium binds troponin-tropomyosin, that rope untwists just a little bit, exposing the myosin binding sites on the actin filament and allowing muscle contraction to take place. And then once the signal to contract is over, the calcium is resorbed into the sarcoplasmic reticulum from the cytoplasm. It's no longer able to bind to troponin-tropomyosin, so it rewinds and blocks the myosin heads from binding, stopping contraction as well. And so what that looks like in an animated form can be seen here, right? So troponin-tropomyosin is blocking sites, calcium comes in, untwists, and now these yellow sites where myosin can bind to actin are exposed. And now myosin heads can bind, and actin can be pulled towards the center of the sarcomere, right? So calcium binds, troponin-tropomyosin exposes the myosin binding sites on actin, and then myosin heads bind, and the muscle can contract. And then when calcium is not bound, that contraction stops, right? And so in addition to ATP and its hydrolysis controlling muscle contraction, calcium has a really important role in regulating muscle contraction as well.